Welcome to Q&A with A&V, everyone. I am Vincent Racaniello, and joining me today from New York is Amy Rosenfeld. Hello, Amy.
Hello, Vincent. How are you today?
Today was a difficult traveling day, difficult commuting day, I should say. But
well, it's the beginning of the holiday season here in New York, so
let me just tell you a quick story. I was waiting on the track. So this morning the trains were all delayed because of switching problems. And this train was coming in the opposite direction. And we know when they're not going to stop, they blow the horn. So it blew the horn and it's motoring through. And all of a sudden it jams on its brakes and you smell the brakes burning. And it misses the platform and he has to back up. And it was a guy on the station. I said, "What's going on?" He said, "He's probably a rookie. He doesn't know what he's doing." Can you imagine? I put my hands in these people every day. Oh my gosh. Anyway, animal crackers. No, not animal crackers. What's the name of the lady that made your painting? Animal what? Do you remember? Animal Party. I wanted to show her this painting of polio virus here. So.
Not as good as my EV68 by Jen.
So it's the capsid with all the VPs, the virion proteins painted individuals.
Who made that for you?
This is made by Eric Carter, who's a grad student at Drexel. And he's here sometimes, although not always. So let's go and see what we have here in the chat.
David Champton gave a super chat. Thank you.
Mr. Ozzycam has a new icon. Look, what do you think it is, Amy?
EVD68?
I don't. Well, it looks like it because it's an enterovirus, right? But it is. I think it's poliovirus, right?
Not the right icon.
Yeah. All right. So there's some discussion of this new variant B1.1.529, which has been found in 10 people, and I think it's absurd that people get all a fuzz over it. There's some scientist in the UK who said, "Well, it has 32 spike changes. It could be a problem." Yeah, and my mother could rise from the dead also.
Oh, really? You're related to Jesus?
No, I'm not.
Every Italian is related to Jesus?
No. That's not true. Someone had a question for you. Let me find it because it had to do with EV68. And I said, when Amy gets here, you should ask her. So let me just find it here. I have to scroll up. Oh, too bad it didn't.
Don't forget to thank the guy who gave you a donation.
All right. So the EVD68 question has disappeared. So please ask it again, okay? And yes, let me thank the man here. Everyone's wishing us happy Thanksgiving. My favorite weekly live stream. And we have our usual moderators. We have Mr. Ozzycam, as I showed you there. We have Vanity Nutrition, who is 34,000 feet.
No, it's 34,000 miles.
No,
she's at 34K miles above Columbus.
Miles above. It's a statement.
Yeah, must be flying. That's great. Happy holidays.
She knows what she's talking about in terms of footage. And then Mr. Les Fabi is here, although I don't have a thing to. Here we go, Les Fabi. Thank you very much for your moderating. Okay, so I just passed a question here. I am curious what research projects you have going on AFM. As a faculty member, I am in awe and thankful of the time you all dedicate. So, Amy, tell them what you're doing with acute flaccid myelitis.
I'm doing a lot with acute flaccid myelitis. Animal models, molecular biology, immune response, viral chimeras. I'm doing a lot.
We have a paper in review, Carlos, which hopefully will be accepted before the end of the year. Then we can talk about it on TWIV.
Yeah. We need to also remind the reviewers that, a week from Friday will be 2 weeks. It would be good if they could get it back before then.
So Carlos, Amy is the lab, Okay, I am just an advisor. I have decided to spend most of my time, doing communication, and Amy is the boss, and I help
Not
her
a
with
good
her.
boss. I'm not a good boss.
So I go to
My
the lab
person.
I change the CO2 tanks, I change the nitrogen tanks, I inoculate the mice, and I bleed the mice.
Yes.
And
After I broke my foot, I was fired from a variety of jobs.
yeah, well, about 4 years ago, she tried to change a CO2 tank. She dropped it on her foot and broke her foot, and she still is suffering. And so I said, you are never touching a CO2 tank again. And she's actually listened. It's remarkable because she's a very strong-minded person. And I think a physical task like that, she's happy to get rid of. The tanks are very big. They're almost as tall as me. They're heavy and unwieldy. They're thin and tall, and so it's easy to knock them over.
Well, a full one was obviously very easy to knock over because it fell on my foot. That took care of that.
All right, so Peter, hello,
yeah.
Peter.
Hello.
60 years after natural measles, does measles memory B cell response fade enough to make someone susceptible to disease? Well, the Panama Islands said that the years, at least 40 years after the initial infection, you don't get disease. I don't know about 60. So if you're a kid and you get measles at 15, then you're 75. I just don't see older people getting measles. What do you think, Amy?
No, I don't think you generally get measles.
I just read something that AstraZeneca may give the longest protection due to better B-T cell. So you don't know, Philip, because we're only out so many months, right? J&J,
Well,
they're both...
I'll actually be a year in December since I was first vaccinated.
That's right. You were vaccinated early because you were in the BSL-3.
Yeah.
Cool.
In theory, we could bleed me and see.
Yeah, but we have other things to do, right?
Yeah.
Anyway,
if
Philip,
I'm going to bleed me, I'm going to take the blood and look at enteros before I'm going to look at SARS-CoV-2.
So the J&J and the AstraZeneca, as you know, are adenovirus vectors, and these are known to induce good T-cell responses. So I think we don't know. We simply have to do the study, and people will do it. Every 2 months, you'll see papers coming out doing that, right?
Yeah.
A&V, can you explain why a second mRNA is needed, but you don't believe a third one is? We've done that a lot, but I suppose we should do it again, Amy.
Oh, okay.
So the second one is needed to fully round out the antibody response. It's not sufficient
Whatever
after the
that
first.
means.
So the problem is we only waited 3-4 weeks between the first and second. If we had gone longer, we might have made a better response. But in the trials, the real protection against COVID didn't come out until after the second dose. And now, after 2 doses, the data show that the mRNA vaccines protect in the 90th percentile against hospitalization and death. 90th
and
percentile.
Severe disease.
And severe disease is defined by the FDA. So I don't see why we need a third dose. So the idea is that now antibody levels are low, but they always go down. They always contract. What's the word I like to use now? I can't even bloody remember.
That is it. They contract.
Contraction. They contract naturally. They always do. And so, yeah, you could get infected because you have low antibodies in your mucosa, but you're going to be protected from severe disease. That's how most vaccines work.
That's how 99.9% of vaccines work, or viral vaccines work.
And Amy sent me an email today, or a text. I get probably 50 texts a day from Amy. She sends papers, and she said, can you believe Fauci's talking about a 4th booster, a 4th dose? And I'm like, this is crazy. What are you going to do? Keep boosting people so you get high antibodies, so you prevent all infection and it really looks good on paper? So that's why we think so. Don't worry, be happy, which I recognize.
It's a song, right? Bobby McFerrin. Don't worry, be happy. I think it's a Bobby McFerrin song from the 80s.
That's pretty good.
Early 90s.
That's pretty good.
But I think that the message is wrong. The message is wrong. So the message should now be, the message should have always been, we're only protecting against severe disease. We're not protecting against infection. We never should have, unless it was a scientific study, started testing in vaccinated people. It was a big mistake, because you have to come to terms that this virus is going to become endemic. It's going to become like a flu. And
yes, it will be.
So you have to learn how to live with it now. And so if you come out and you say, vaccines are to protect against severe disease, not mild disease or infection or exposure, then you stop. Then you just go, it's 2 doses, you space them out now that at least one of them is officially approved, and you do it the right way, and then you just move on. Because all you need to know is that even if cases go up, hospitalizations do not. I mean, unfortunately, that's not what we're seeing in Massachusetts, and another state that escapes me off of the top of my head. But for the majority, like if you listen to Daniel, it's hospitalizations were at 4,000 as they were in April, or cases are at 4,000 as they were in April. And instead of 1,500 people being in the hospital, now you have 30. That's all you needed to do.
Okay. Thank you, David, for your contribution. Really appreciate it. All right. You say that T-cell assays are hard to do, but also repeatedly say the epitopes aren't changing. How do you know? Well,
so Shane Crotty's papers.
Shane Crotty and Alessandro Sette, they've both been on Twitter to talk about this, and they've both published this. You take lymphocytes from people, you put them in culture in the lab, and then you put in peptides that are from the virus, and you see if the T cells respond to the peptides. And if you put variant or ancestral virus peptides in, they respond. I mean, Ellis, in his latest chat, Sette said a few of the T-cell epitopes change, but it makes no difference. You still have wonderful recognition of the peptides by the T-cell receptors.
There's a paper. He and Crotty are on Farber's paper that you're going to go listen to for the defense, where they come out and they call it immune memory. Tissue relevant immune memory. It's in Science Translational Medicine.
Okay.
So
good for them.
Yeah.
Hello, Lost Laboratory.
Well, it's not about the paper and where it went. It's about the finding.
Right.
So good for them is not the right response. It's about the finding, which is that immune memory. We will have immune memory and it's fine.
Yeah. Well, we've done some other studies that have concluded that as well.
Right.
T-cell and B-cell memory.
Right.
Hello, Lost Laboratory made a nice collection of T-shirts. I'll have to review them. I'm going to show them to you, Amy. They're very cool.
That's great.
Mike, there is now a nasal vaccine to prevent Alzheimer's? I doubt it.
Yeah, no.
There's no vaccine because, as you say, how would you remove plaques? No.
There is no.
There's no vaccine. We still have nothing for Alzheimer's.
Except some crappy drug that doesn't really work. It got everybody's hopes up, and then all these people quit.
Any new understanding of the cause of loss of smell and taste? No. We did a paper recently on TWIV, right, where they took out the nasal epithelium from people who just died. And the first sentence in the paper, 11 months after we first discovered this, we still don't understand what's going on. So
it's a very hard thing to study because you can't really model. It's very hard to model in animals. You would need to have somebody like Axel do the modeling.
Yeah.
Because he's really understands the olfactory bulbs and stuff.
Do you think it would be interesting to talk to him and say, what do you think is going on?
Yeah, I think he might have an interesting perspective.
He might say, you're the virologist, Vinny. What do you think?
But this isn't a virus problem. This is a neurotransmission problem.
That's true. All right, here's the question. I'm curious if you know any innovative, effective, best-in-class methods to survey for 68, perhaps in Wales.
Um, so they have done some wastewater studies in Great Britain. I'm not sure if it was in Wales or not. Um, but so I have a little bit of a, I think that that's great. But I do have a little bit of a problem with wastewater studies for a respiratory virus because that gives an opening to a certain lab that wrote a paper that is now going to screw up the field for the next 40 years in the vein of Flexner that is suggesting that EVD68 is a, evolving into a gut pathogen. No. Um, so I think probably the best way to do it is probably if we could take advantage of like the flu networks that they have, except make them the opposite season, because the flu networks really go from like October to March, right? And we need to go from like May to November.
All right. What's your best guess as to the long-term durability of the vaccine? The Crotty said last year, like many years,
Maybe 10
right?
years.
10 years?
Maybe 10 years.
Spiffy, you're going to be good. And all this booster is going to be forgotten. You'll see. Well, we don't want to predict, but that's what we think. Any last-minute advice for PhD applications?
Don't do it.
No. That depends what you want to do. Do you want to do virology? I don't know. Do you have any? If it's, let's assume it's virology because he's here on the Virology live stream. What would you suggest?
Like what advice for what? How to fill it
Where to go?
out?
No, I'm sure he can fill it out. Where to go? Where to go?
Or what programs to go to?
Yeah. Yep.
I would go Wisconsin, Chapel Hill, Texas and Galveston, WashU,
Chicago?
no.
San Francisco?
Sinai, no.
Mount Sinai? No San Francisco? No UCSF?
No, because really it's just Raoul.
Okay.
I mean, Raoul is great, but you need to be able to rotate through more than one laboratory. I mean, Corette and Carla and Peter at Stanford, but it's also still small. And Gins and Rugman is there. So it's good. I mean, it's good. But I would say Wisconsin, Chapel Hill, Texas, Galveston, Sinai, and yeah, maybe Penn with Susan and Bates and other people. Yeah, those 5.
So Rugman, for those of you who are at the Virology Live today, Rugman is Harry Greenberg, who's a gastroenterologist / virologist at
I can't always remember his last name.
Stanford, and he has a wonderful Oriental rug collection. That's why Amy calls him that. But he gave me the slide. He gave me the slide of all the water going over Victoria Falls.
Yeah, he's very lovely.
Oh, look. Happy Thanksgiving, Amy.
Happy Thanksgiving to her, too. Flying high.
Dame Sarah Gilbert thinks boosters might not be necessary after all. Well, that's lovely. I'm sure other people agree with us as well. It's not just us, because people say, oh, all these other experts think the opposite. Yeah, well, we can find some experts who don't, and it's not a numbers game. It's just, in the end, what works out. What are the factors driving the increase of COVID-19 cases in Germany and France?
The non-vaccinated going indoors.
40% of the population is not vaccinated. I think that's the number.
I don't know what the number
That's a lot of people. And people were just, chilling there, back to life. They're getting together. It's going to happen unless you get more people vaccinated. Hello, Charlotte. I emailed you with Amy's address so you can send her the painting, the abstract art that you would like to send to her. Thank you very much. Bamlanivimab doesn't neutralize Delta for this reason, was pulled for use on its own. So why is it in the cocktail? It seems wrong-headed. What am I missing?
Well, there's other things out there besides Delta. And I don't know that it completely didn't neutralize. It might just not have neutralized as well as others.
I think it has to do with licensure also. It's part of the cocktail. They can't just pull it.
Yeah, I don't know.
And as
I don't
Amy
know
would
enough
say,
about it.
as Amy would say, neutralization is not the whole story.
It is not.
There can be other non-neutralizing functions of antibodies that are important.
Yes.
I hear there are 10 to the 31 viruses in the world. There are 10 to the 31 bacteriophages in the waters of the world. That's just viruses of bacteria. They're just particles. In fact, they were counted by staining seawater with a dye that stains the nucleic acid, and it's just counted. So it's not strains, no. It's a lot.
It doesn't account for anything that infects animals. Or I'm sure like coral reefs have viruses and all that other stuff that lives at the bottom of the sea. So, I mean, that would have been kind of a cool graduate project, is to figure out if coral reefs have viruses.
Yeah, they did that in Forrest Rower's lab, among others. Yeah. So DG wants to know, what should we be measuring to determine what policies should be in place? Positivity, cases, death. So Amy and I were just chatting in the pre-show, which means nobody hears it except us. And she said we need to stop testing vaccinated people, right?
We don't test anybody vaccinated for whether or not you're exposed to flu or to measles or to polio or to Hep B or to HPV or any of that.
Yeah, I think it's painting a false picture because we don't know what the positivity means.
Well, it's a single time point. So, yeah, I mean, you have no idea. If you want to do a scientific study, that's one thing. If you want to use it as an epidemiological something or other, it's a completely different thing. It's the same argument I have about sequencing. If you want to do a scientific study and say, we're watching the virus adapt to a new host, that's one thing. If you want to scare the bejesus out of people, that's a separate issue. And I don't agree with that issue.
So when in the old days, when we give the measles vaccine, all we do is look for measles. That's it. We don't measure who's got virus, this and that. Even
when you gave polio, you didn't measure who got who got fever and stuff. You just measured who got paralysis.
It's not helpful.
So
you just want to know about disease. Yes.
Right. So people should have stopped. That's all that they needed to do. But it's not in they obviously need to go back and look at the polio literature, just like we did, or I've been doing for a variety of work, right? To find out that we're repeating all of the same mistakes in various fields.
I think you need to look at, if you want to determine policies, right? What the policy you're going to determine. Are you going to shut things down? Or are you going to shut schools? Or are you going to make people mask and so forth? You need to look at positivity rates. Amy would say the percentage is the most important, not the number of cases, right?
Yeah, I don't care about the case numbers. I only care about positive rates.
And I say you look at deaths. Yes, go ahead.
Right.
I'm sorry.
I mean, like, it bothered me that they would say, oh, we've tested more people than Seoul, Korea. Yeah, well, Seoul, Korea, that percentage was, 80% of the population. Here it's like 4%. You haven't tested anybody. I mean, people don't understand how to analyze data. It's very upsetting.
Oh, Charlotte
should all just give back they should all just give back their degrees and start over again.
Charlotte says she would donate a painting. We could auction it to raise money. That would be cool. Yeah,
that's great.
We could do that.
Sure, for sure.
Thank you very much for your contribution, Phoebe. Not sure how to pronounce it. What's the function of ACE2 and why is it such an Achilles heel? Well, ACE2, what is it? It's a regulator of the renin-angiotensin system, which is involved in blood pressure, right?
Yes.
And so SARS-CoV-2 has to bind to it to get into cells. So if you can block the binding, that's a Achilles heel. For most viruses, it's the same.
Yeah.
Would it make sense to create an mRNA vaccine coding for both spike and nucleocapsid protein?
Sure. Go ahead.
Then you would. So the thing is that Amy and I have talked about this before. Certainly, some antibodies against spike can neutralize infectivity, right? Can block infection of cells. The question is, can antibodies against N do that? They might. Some of them might. But there would certainly be T-cell epitopes.
But they'd have to like, would they be like AD, which neutralizes internally?
Could be. I think that would be really
interesting. That's very cool.
Is there a test that can detect our memory cell response? So memory cells, not antibody or interferon.
Yeah, you can harvest memory B cells from the bone marrow. It's not comfortable.
You can do that from the blood also, so it's not as good. And you have certain markers on them, right, Amy, that distinguish them as memory cells.
Yeah, for sure.
Happy Turkey from Central Valley. That sounds like wine country. Is it wine country? No. It's a hot place, right? Okay.
I think so. But Turkey Day is never happy because nobody likes turkey. Turkey is
it's 35,000 feet. See, 6.5 miles. 35,000 feet would be out of Earth, right? You'd be out of orbit, I think.
Yeah.
Oh, look, Jean's child was hospitalized with EV D68 in 2014, but she recovered.
Oh, she is a she should write to us. We need to get her email because she can donate samples.
Just blood.
Yeah, just blood. And it would help us understand where we're going. That would be, and if she has a sibling that also was infected but not developed severe disease, that would be really great. Get her information Secretary.
Do we have an IRB for this?
Yeah, we do.
Okay, so just if you're interested, just collecting blood would be done locally, and we'd like to get B cells and see what the antibodies they make.
Yeah,
Vincent
B
at
cells
microbe.
B cells and T cells.
Just once, Vincent at microbe.tv, Gene, if you're interested.
That would
anyone
be
else
super
who
helpful.
Anyone else who has kids that have had EVD68. So 2014 was the first big outbreak year in the U.S. Then there was 2016 and 2018. So get in touch, and we'd love to have some blood.
That would be super helpful for our studies. I'm so glad that she recovered just fine. But yeah, it's very scary. Like, I'm reading the Oshansky. Is that his name? Oshansky?
Oshinsky.
Yeah,
David Oshinsky.
The polio book. And like all these, we're just repeating the history of polio from the 30s and 40s. And it's very upsetting. And so, yeah, we need to fix this.
What's going on with Novavax? Are many people holding out?
I don't know if people are holding out, but good luck to you. They seem to be having problems.
Well, it got an EUA in India, right? So I guess it's imminent. But, yeah, I'm not sure. People say a lot of holdouts will take it, but I'm not so sure about that. I think that's wishful thinking.
Mm-hmm.
How would you set up an animal model to test the hypothesis that COVID increases the chance of Alzheimer's?
There is no animal model for Alzheimer's. That's the problem.
There are those transgenic mice with...
they're not good enough.
They develop plaques. Yeah, they're not great, but that's all we have. So what you'd have to do is you'd take something like that and infect them and then see if
you'd
the
have
mice...
to do a variety of behavioral tests.
You do behavioral, but then you can look in the brain and see if you get more plaque formation.
Yeah, I don't think that this is a good line of research.
No, we could answer the question, right?
Yeah, I just don't think it's a good line of research.
Prior infection plus vaccinated. Should I even consider a booster?
Don't need to. Not at all. It's fine.
I don't think so. Okay, here's a little thing that's been going through the pre-show comments. Can the virus mutate itself out of existence? I heard that's a theory on what happened in Japan.
I don't believe it mutated itself out of Japan. I believe that Japan still has cases. So I'm not
It's
clear
there.
where what?
It's there. I'm sure it's there.
I'm sure it's there too. So I don't know what it means that it mutated it out of existence.
So the
Doesn't
theory
mutate
is,
it out of existence.
which is being pushed by a Japanese geneticist and which is just going nuts in the press, is that he said it became so good at moving through the population that it's going to infect everyone and then it can't infect anyone else. And that's just bogus. There's always hosts left. Yeah, no, the birth rate in the world, for example. So no.
My brain hurts.
And influenza, rotavirus, norovirus, enterovirus D68, they have new hosts every year. They cause new infections. They're always around.
Yeah,
Viruses are very amazing, much more amazing than mutating themselves out of existence.
Yeah, they're not mutating themselves out of existence.
Would Novavax potentially have a lower side effect? It probably has the same side effects. Pain at the injection site, which is the main one. Muscle soreness, fatigue, fever, right? You have all those things, right, Amy?
Yeah.
I'm sure they would have the same. Novavax has an adjuvant.
I was going to say, but I get them for almost every vaccine. I get, like, my arm, when we go for our annual flu shots, usually you say you're fine the next day. I usually say my arm hurts for like 3 to 5 days later.
Yeah.
But I'm kind of what do you say? I'm kind of what did you call me the other day? Somebody just said that I was physically, like, even though I run a lot and stuff, I'm not like very large person.
You're frail?
No, they didn't use the word I was frail, but they were like, yeah, I could see how you could have that happen to you. There's not a lot of meat on your bones.
Karen hated changing tanks. I really enjoy it. I love spinning them down the hallway.
Yeah, he does.
Amy
he likes
looks
to show
at me.
off. He likes to show off.
I've never shown off a day in my life.
Sure. Keep telling yourself that.
So Lucky Six has said to Peter, I bet we get reinfected with measles through life and get boosted. Possibly, but in the U.S.
Obviously it has to be an aborted infection.
Yes.
Every active infection results in the rash, and we don't get rashes.
So it depends where you are, because in some places there's not a lot of measles virus circulating, right? But in many parts of the world there's a ton. Brazil has a lot, for example, many other countries.
Well, yeah, I mean it's endemic in other areas, not here.
Concerned about the effects after possible 4th or 5th doses. Could you expand on the possible side effects?
I don't think that there are going to be additional side effects than what you normally get when you get a vaccine. But I just don't think that it's a good public health policy.
Yes, and if it's not, if the only point is to raise antibodies so that you just block infection for 6 months, that's not really good, because that's not what we do with all other vaccines, as Amy has said.
Well, I don't think it's a sustainable policy whatsoever. So I just don't think it's the right thing to do. All right. It's like 8:35. I have about like 10 minutes more. Then it's time for me to go home for the holiday.
How does molecular iodine work as an antiviral? Is it
I
at
didn't
all?
know it was an antiviral.
I don't. We have, between Amy and myself.
I've never heard of it as an antiviral.
Okay, so here's a paper in a journal, Amy, called Medical Hypotheses. The antiviral action of povidone iodine may also have therapeutic potential. So we don't know the mechanism.
I could have a paper in that journal called of medical hypotheses. My hypothesis is the blah blah blah. If we published articles on every idea and every hypothesis I have, we'd be like publishing a paper a week.
Unfortunately, we couldn't have run longer trials. Yes, of course. That's why they were sped up. They were purposely designed to be sped up so that we would get the information we needed, but perhaps not have the distance between boosts. Yeah. In your TWIV on Entero 68, look at Amy. A lot of questions on EVD 68. You said it returns in 2-year
fantastic
cycle, but has skipped a year. If Europe not skipped a year, it means now out of step with the USA. Will they eventually resynchronize?
So generally they do synchronize. I don't know that I would say that Europe did not skip a year. So 136 cases is generally not a lot of EVD 68. It's not a lot of entero cases. And you know, we're completely a passive surveillance system. So why we put out calls for people to look out for it. If you don't have the AFM, it might not be on your radar, even though we ask you to look out. It's really up to the state and the district health departments. So yeah, they resynchronize, and basically the same strains or variants are from each other, but it also goes back and forth. So you can see some trafficking between the isolates between the different countries.
Oh, thanks, Amy, for your truth.
Oh, it's good.
Is good T-cell response with an ad vector a bait and switch where T-cells show up expecting a multi-course ad meal and instead get spiky breadsticks? Yes. What?
No, ad has a spike. Ad has a spike. It's called the fiber.
Yeah, you get adenovirus T-cells too, okay? But
yeah, but isn't the fiber of adenovirus isn't the gene of ad completely replaced a spike? And fiber is the receptor, is like a spike.
No, but you don't replace spike with spike. No, you don't replace fiber with spike. No.
Oh, really?
No. You just add it. You delete like E3 or E4 or whatever.
Why don't you just replace fiber with spike?
Because you need fiber to get into the cells, right?
Why? Why can't? Oh.
Spike is a transmembrane protein. Fiber is stuck in the capsid there. It would be hard to engineer. Not impossible, though. It would be interesting, actually.
I don't know. It would be. I would have thought we could.
You know, Amy, you could just the tip of the fiber, the knob.
Yeah,
you could replace that with the RBD.
Yeah, exactly.
Yeah, that would be good.
I don't know why we. Yeah.
Oh, hello from Finland. My colleagues have third doses. I am fine with 2. Yeah, you'll find people fine with 2 everywhere. Yeah. I find it obnoxious. I'm standing at the train station today, and there's a big poster of a family eating turkey dinner saying, "This holiday, get a boost." I just,
Yeah, I understand, but they're not going to believe us over Fauci, so
I get it. I get it. It's no problem. For age 50 plus and comorbidity, what's the literature about hospitalization and death for double vaccinated?
It's significantly reduced, like almost 10 to 50-fold.
Hospitalization and death? You mean for vaccinated, for being vaccinated, right?
Yeah, 2 vaccinated, and if you're over 50 in comorbidity, it's like it's like between a 10 and 50-fold decrease risk.
Yeah. So you're 90-some% protected against hospitalization and death, right?
Yeah, something like that, yeah.
Now, someone had told me that at the latest FDA advisory committee meeting, they said they have some data that these numbers are starting to decline, the protection, but they're very small.
I haven't seen that.
But I just think it's
But
noise.
it is small. I mean, the truth of the matter is you can make statistics say whatever you want. So I don't know what small means.
When the virus is endemic, is it still just as bad for infants as soon as they stop breastfeeding?
Well, it could become a new polio, right? Where you cleaned it all up, you stopped breastfeeding, and then children got exposed, right? And that's why polio was called the infantile paralysis, right? So you could imagine that if you can imagine, it's not going to be bad for them. I mean, they're not going to develop severe disease, but they will get infected, and they will become, potentially sick.
But not many babies get sick. I mean, they do, but the rate is much lower for reasons that
Well,
are
it
not clear,
depends on
right?
it depends on what you're calling sick. They don't get hospitalized with ARDS,
Right,
but they
right,
do get sick. They get a cold.
right. And then they will
So
have immunity, right?
right. So to say that they don't get sick is not correct. They don't get ARDS.
Yeah, I meant severely ill.
Yeah.
But even sick is lower frequency than higher ages, right?
It's a cold. How do I know if I don't take you to the doctor and get a test, how do
Okay.
I know what caused the
Yeah.
cold?
Okay. Okay.
I don't know.
Yeah.
I mean, there's 400,000 cold viruses. Every time you turn around, we're identifying a new cold virus.
Are there
I don't
10 to
see.
the 31, Amy?
Yes, there's 10 to the 31. Yeah.
Thank you, S. Son, for your contribution. Really appreciate it. Amy, I'm not sure how the message switched from prevent disease, which somehow go memory hold. Yeah, I don't know. We forgot all of our basic immunology.
That's above my pay grade. I'm not in charge.
Hey, look at this. EVD68 first isolated in California. AIDS first reported in California. That's right.
That is true. And EVA71, also from California. Yeah.
Is A394V the virus kill switch?
I don't know what A394V is.
I don't know. I'm V and you're A. Maybe this has something to do with us.
I
Could
don't
it
know.
possibly be? Let's type it in. A394V. No, it's another change in spike. It is in the Delta variant. Yeah.
No, it's not a kill switch.
It's not a kill switch. No.
Okay, like two more and then
So if I get the booster, I'm still not protected against infection. Well, in 6 months you won't be. You will be as soon as you get it for 6 to 8 months, and then your antibody levels will contract, and then you can be infected
I don't
again.
know if 6 to 8 months is the right time frame, but yeah.
What are you thinking?
I think the antibodies spike for like the initial 3 months after the booster, and then they gradually contract, and you'll be back down to the same level that you were. So if I titered you today, gave you a booster tomorrow, and you came back in 6 months, you'd be at the same level that you were today. So I don't see the point in this. I don't think we should ever. I think we should never have aimed. We didn't actually aim for protection against infection. They did the first test too soon, and they misinterpreted the data, especially the Israelis, and therefore there was no walking it back. And now we're stuck with this, which is very, very unfortunate.
Steph likes your office, Amy.
Oh, that's great. Unfortunately, it's not my office. It's Vincent's.
Thank you, Steph. I'm glad you like
So
the art.
here, what am I? So Marie Elster says, you are wrong, Amy, in big capital letters. So I want to know what I'm wrong about.
Doesn't matter. She's gone.
I just want to know what I'm wrong about.
No, they just want to be nasty.
I was going to say, before you start saying that I'm wrong, you should just tell me, yeah.
You know what Daniel's quote today was?
I don't know.
We can disagree without being disagreeable.
Exactly.
Do you know who said that?
Sounds like Martin Luther King.
It was RBG.
Oh, yeah, could be.
Taylor, it doesn't matter if you can't donate. We will do this. The assumption is the people who can donate will, and that'll make it possible for everyone to hear this. That's our philosophy. You got it. How are you? You want to leave in 1 minute?
Well, we can do two more, and then I have to go.
So can you help me with measurements? Pfizer dose is 0.3 ml, 30 micrograms. Moderna is 0.5, 100. Shouldn't the Moderna equal 50? Well,
no. So volume and weight have nothing to do with each other. So you could have so mercury and water are a good example. So, 1 milliliter of water is 1 gram. And I think 1 milliliter of mercury is 13.4 grams. Because it's the density, I believe, or the molecular. Yeah, I think it's the density. I think that's right. So they don't really have anything to do with it. So you actually don't really care about milligrams and milliliters. You care about molecules. And since you don't know the molecular weight of what's in there, you can't really say that they're all different. They could be exactly the same.
So the lady who said you were wrong said antibodies wane after 3 months, and that's not right, as you know. Maybe monoclonals do.
Yeah,
Depends on the
monoclonals.
monoclonal.
Some monoclonals don't.
So Marie, be civilized. Just say, I think they wane in 3 months, or I would say contract in 3 months, and then Amy would say, oh no, those are monoclonals you're thinking about, not capital letters. You are wrong, Amy. Come on, be civilized. Ay ay ay.
So yeah, so you don't go. So I think it was again another misunderstanding of science that scientists talk about molecules and moles, not weight.
So 68 California, AIDS California must have been an equivalent of Wuhan lab at UC using screwball logic, right? Yeah, it's screwball logic, right? Just because it's where you first found it doesn't mean it. Yeah, AIDS was originated in Cameroon, right?
Yeah. Well, actually, AIDS was, yeah. I mean, the first huge outbreak was in California, but when they backtraced, there actually were cases in Haiti first. Yeah. I mean, you'd have to say that AIDS came from the LAX, right? HIV came from LAX because that's where all the people came from.
All right. I'm obese, 80, have high blood pressure, sleep apnea. I'm protected with 2 doses, right? Maybe not, because not everyone's protected even with 2, right? It's only in the 90th percentile. But I'm not clear that a booster is going to give you that protection. That's what the assumption is by the CDC and Tony Fauci, but I'm not sure about that. I know people with comorbidities who have died. They're 2 doses, they get sick, they go in the hospital, they die, because no vaccine is 100% protective. So will a booster take care of that? That's what we don't know. And that's what no one can tell you is the case. Is that fair, Amy?
Yeah, that's fair. All right. It's time for
one
me
more.
to go.
This one. Here, one more. Look, thank you, Amy, for that explanation of how vaccines work. I wish politicians would listen to you. I'm afraid Dr. Fauci needs to retire.
I don't know if I would go that far. Politicians aren't paid, and that's not their skill set. They're supposed to know who to hire that has this, who is the experts in the field. And they're supposed to be able to discern who's the bullshitter and who's not. Unfortunately, they're too consumed with making sure everybody that they get their own way and everybody conforms. It's not a good plan. What do you think?
As we have discussed, that many things were misstated and mismanaged, and it reduces confidence in certain people so that they're no longer effective.
Right. But, politicians also it's their job, and they only care about getting reelected.
Yes,
and
well,
so
that's it. Yes.
They push their point of view, and you use, we have a lot of scare tactics now. We're very good at
it. And they're not scientists, and they're not really interested. They're interested in getting reelected, right?
Yes.
Thank you, Amy.
You're welcome. Happy Thanksgiving.
Have a good time in New Jersey.
I have to go look at turkey. I don't like turkey.
You're having turkey?
I was told that there is no such thing as a Thanksgiving without turkey. That is what my sister and my mother told me.
I'm sorry.
I am no longer, I am one. I am going to lose this argument. So it's just the way it is.
Sorry, we're not having turkey, no.
What are you having?
Actually, I don't really know.
Well, it's tomorrow, so you better be having something. Well, I know that
we're having eggplant parm.
Yeah, but that's only the appetizer.
I don't know. I actually don't really know. Obviously, I've been more interested in science communication, which is bad, right?
I don't know. You have to ask if that's good or bad.
I'll see you Friday, okay? I'll be there to change the CO2. Yeah?
Yeah, I got it. Yeah. Yeah, I know.
Mice.
I know.
Okay.
I was thinking if we actually really needed to look at the CO2 and liquid nitrogen.
But
But
we need to do Mice Friday, right?
absolutely. We have
All
lots
right.
to do Friday.
See you
All
then.
right.
Happy
Bye.
turkey. Bye.
Happy turkey.
Okie dokie, folks. Dr. Griffin, Daniel Griffin, recommends monoclonals for double vaccinated at risk. Yes, he does. If you have high risks, like the lady just said, 80, obesity, etc., you get infected, you're tested, you get positive tests, he recommends monoclonals, yeah. So that would be an alternative to getting a third dose. But they don't last forever, right? You'd have to get, if you did happen to get infected again, you'd need more. But that's not a big deal. Do you believe that immunosenescence may be why it's most elderly and immune-limited vaccinated that are in the hospital after vaccinated? Well, I think that's part of it, right? There are certainly some people who don't respond well, and the older you get, the less well you respond because, yes, of immunosenescence. That's absolutely correct. But also some people with apparently normal immune systems also do not respond. And so, you can't expect any vaccine to be 100% protecting against severe disease. In fact, if you remember, before we had vaccines for this virus, the FDA had said we'd be happy anything 50% effective or better we're happy with. And unfortunately, we got 90 percentile for a couple of vaccines. And then as the immune response contracted, people started freaking out that the overall efficacy against any COVID declined. And that's simply not a worry at all. That's where we are today. Thank you, Brittany, for your contribution. How long to wait before the first two vaccines? So here's the thing. After one dose, you don't get maximal protection against any kind of COVID. That's what the first trials told us. So it's good to get that second dose in. But I would say not less than 2 months, because that's a good amount of time to let the antibody response mature. If it were my kid, that's what I would do. But you should see what they're recommending and go with that. If the child is, especially among people who are often, in school situations where there is a potential of getting infected, you might want to get it sooner. Oh, you didn't see the picture? Here, I'll show it to you. So those are the 4 capsid proteins of polio. There's poliovirus in the middle. And around it are the 4 capsid proteins. So this is painted based on the structures that you generate on a computer. You can see the nice 5-fold and 3-fold axis there. Look at that. 5-fold axis, it's a star, right? And then here's the 3-fold axis. And then next to it is the 2-fold axis right there. It's very nice. So I'm going to put this up in the incubator. Going to have a lot of art in the incubator. It's cool. Weakened and activated vaccine versus mRNA in healthy people. Well, the inactivated vaccine that was developed in India, is it Covaxin? We just reviewed the data. It looks pretty good versus severe disease and hospitalization. I think it's fine. But we decided not to do that here in this country. Antibody breakthrough in mice could lead to a vaccine. All right, so that's the headline. There is no vaccine. It could lead to a vaccine, and I don't know what the antibody breakthrough is. I'll have to take a look at it, folks, and see what this is. Let's see. I can do it really quickly right here. Let's get a new window. Alzheimer's result. That's the query. News. I guess we should look at news, right? One of you could send me a link. Is a nasal vaccine on the horizon? Let's see what they have to say about that. Damn cookies. Phase 1 trial of a nasal vaccine is beginning. So what is the immunogen that they're doing in this phase 1 trial? And the director is Howard Weiner, who I know. Protolin to stimulate the immune system. All right, so that's an adjuvant. Why can't they tell us the antigen in the first 5 paragraphs of the bloody article? Don't people want to know what the antigen is? Not saying. I'd have to go look it up further. So sorry, I don't know what it is. But it's a phase 1, so it's not a vaccine yet, right? It's not a vaccine. Why is measles highly transmissible? Well, there are 2 answers to that. One is that we actually don't know. Nobody has really understood why it is so transmissible, but I can tell you what the hypotheses are. And part of it is that the transmission seems to occur in all droplets, respiratory droplets of all different sizes, including big ones and intermediate-sized ones and really small ones that travel long distances. So the small ones can remain suspended for a large period of times, travel over 100 feet, so potential there to infect many people. And that doesn't happen as often with SARS-CoV-2, although there are people out there who say it does. I don't think the data show that. The smaller the droplets, there's less virus in it. So I don't think there's enough SARS-CoV-2 in that to infect someone, unless you're in a room, say, with someone exhaling SARS-CoV-2 for a long time, and then you're continually inhaling enough to get an infectious dose. But with measles, apparently the amount of virus in those small droplets may be enough. That's my speculation, but we actually don't know. Can you provide an overview of the 2 types of rapid at-home tests? So as you say, there's an antigen and an antibody test. So in both cases, you can get them in lateral flow devices. They're little cassettes, kind of like a pregnancy test, where you're looking for, in the case of pregnancy, it's an antigen test. And so if you do a nasopharyngeal swab and you apply it to the lateral flow device, if there is virus protein in there or antigen, the device itself has antibodies that will pick it up and make a line. And it has controls built into it so you know the assay is working, and also will look for different types of antibody that'll tell you you're probably recently infected or you're infected over 6 months ago. So that is, I'm sorry, I'm confusing the 2. So the antigen test is just looking for one kind of a viral protein. It can be either the spike or the nuclear protein. They can be combined so that you can distinguish between vaccination, which would just give you spike protein. Forget that. You're looking for antigen, viral antigen in the nasopharyngeal swab. Could be spike, could be nucleocapsid protein. And that indicates an ongoing infection, okay? Because you have enough protein to activate those tests, to give you a positive in those tests. You probably have a good amount of virus in you, as opposed to a PCR, which is not an at-home test, but it's very sensitive and is not necessarily giving you a positive when there's it can give you a positive when there's small amount of viral RNA present. So that's an antigen test. The antibody test is not going to tell you that you're currently infected. It will tell you if you have been at some time, because it takes weeks to develop antibodies, by which time your infection is probably over. And it's the same kind of format. You will look for antibodies against spike and nucleocapsid, and they can distinguish between, if you were just vaccinated, which you would just have antibodies to spike, or if you've been infected, you should have antibodies to the nucleocapsid protein. So, and you could be both infected and vaccinated, in which case you would, again, have nucleocapsid protein. So that's how they work. And so the antigen is a good rapid test to do every couple of days if you are in a situation where you think you might be exposed, right? Whereas an antibody test will just tell you if in the past you've responded to a vaccine or if you've been infected. But the problem is the number you get on the lateral flow, you're not going to get a number. You're just going to get a yes or no, right? And there are lab-based tests, clinical lab-based tests that can give you a number, but we don't know what that means in terms of protection. So I'm sorry to mix it up a little bit there. I thought I saw in one of your slides immunity from COVID is 1 year because it's mucosal. Is that correct? And no equivalency in that case with the vaccine? I don't think I said that about COVID. So far, mucosal immunity hasn't been studied all that much in terms of longevity. But certainly, you can find studies that look at serum antibodies and B cells and T cells, and they go out to a year now, yes. And as we mentioned earlier, Crotty and others have predicted that could be many years, 5 to 10 years, comparing that with other vaccines. So I think this immunity is more durable than we think. The fact that antibody levels are contracting is really not a bad sign. That's the way it happens with antibody responses. Give some attention to the rationale in Israel, U.S. for boosters. Even if you don't agree, a 60-year-old chair of medicine doesn't want to be knocked out with even a mild infection. That's fine. It's your choice. I'm not telling you what to do. You ask me whether a booster is a good idea. I'm telling you what I think. I'm not telling you what to do. However, what are you going to do in 8 months when your antibody levels have contracted? You're going to get another booster? You're going to do that the rest of your life? It's not a good public health plan. If the vaccines aren't good enough to do what you want, then redesign them. But as Amy said, they're originally designed to prevent COVID, not to prevent infection. And so we're changing the bar, and that's not appropriate. And Daniel Griffin has said the same thing. He said, I don't want any kind of COVID, so I'll get a boost. Good. Go for it. I mean, I have a friend who had 2 doses of Moderna. He got infected. He said, if I didn't know, if I hadn't been tested, I would have thought it was a common cold. And that's the problem with testing vaccinated people, that we are getting too much information that we don't know what to do with. From what I see, Michigan is reporting 25% hospitalizations and death in fully vaccinated people. I don't know. I don't know where these data are coming from. One of the problems is that hospitalization can vary, right? The bar for hospitalization can vary. And as Amy said, in Massachusetts, there seems to be an increase in deaths. We don't know what's going on there. And so I can't answer your question. I think it's highly unusual because if you look at the major studies that have been done and published by CDC and others, 90th percentile protection against hospitalization and death. So we're talking about a regional difference. Could be anything. I'm not sure. But we'll certainly get some solutions in the coming months, hopefully. I was in outdoor contact with my friend's son 4 days ago. I'm double vaxxed. He's not. He had positive rapid tests today. His mom, other 2 siblings, all negative. What should I do? Well, you're double vaxxed. You're good. You don't have to do anything. About yourself, I presume you're asking about. And remember, not everyone's a transmitter. Only 80% of transmissions are done by 20% of people, so that may be the end of the transmission there. But you're okay, I think. I don't think you have a problem. If you want, get an antigen test if you're worried. If you have a comorbidity and you're worried, get an antigen test, and if you're positive, they'll give you monoclonals. The days you could call and ask to speak with your doctor. Well, some people can still do that. You should be able to. If you can't, you should get a new doctor. Because they should be able to talk to you, or at least talk to their physician assistant or nursing assistant for sure. And if they're concerned, they can raise it up. I can speak about Germany, large parties and bars. So this is the thing that you never read in the papers. Oh, infections are going up. We don't know why. It must be a variant. Large parties and bars. And Ron Fouchier said that on a TWIV not too long ago. Yeah, we had a lot of parties. Why can't we be honest about what's going on? I disagree with your Alzheimer's treatment predictions, which is what? That we don't have any, or we certainly don't have any. The beta amyloid drugs didn't work because they're given too late. That's right. We need a 50-year trial. My generation will find out. That could be. There are many companies with drugs. And what you do is you get people who have the early signs, right? And you enroll them. And you see if they progress. And you can do that not in 50 years, but in a couple of years. And so I know this because my wife works on an Alzheimer's drug. Okay, so don't think I'm just pulling it out of my ear, all right? There are trials ongoing with anti-herpes antivirals because some people think herpes virus is a major stimulant of Alzheimer's. So they enrolled people with early onset. And I know this because I know a few people who were declined because they were too far advanced. And they're going to treat them for 2 years and see if it makes a difference. And you have to have repeated MRIs to do that. Not a 50-year trial, no. Sorry. Yeah, Sarah Gilbert is the adeno vaccine leader at Oxford, and I did have her on TWIV. Please go listen. She was really good. You know, not many people listen to TWIV, but it's a damn good show. And we get good guests from time to time. And when we don't, we have cool conversations. I'm sorry more people don't listen. They're scared because it's 2 hours. But you're happy to go to a 2-hour concert, right? In Colorado, 18% of hospitalized were vaccinated. Yeah, Massachusetts is in Colorado. But as Daniel has said, is in New York, that's not the case. So I don't know why the difference. And you have to think about reporting. I don't know what's going on and if the hospitalization is different. But, of course, death is pretty much the same. So that's got to be the bottom line you have to look at. Can we get the record straight? When someone says that, it usually means there's no answer to your question. Why do some people have severe reactions and some have none? And does the more severe reaction infer a more robust immune response? So the last part, no, it does not. There's no correlation between a reaction to a vaccine and a robust response. You can go look at the data for the trials, the phase 1 and phase 2, phase 3. They all have side effects and antibody levels, and you could correlate them. If you look at the FDA submission, they will give it on a patient-by-patient basis. Now, why is this? Some people have everyone has a slightly different immune system, and what is seen by your vaccine is the innate immune system initially, and it reacts very quickly within hours. And some people have really robust interferon responses, and some don't. You know, the interferon response is also regulated by specific proteins, and some people can't regulate it as well as others. I think these are all possibilities, but it's hard to study because there are so many people in both categories, so it's hard to know, even if you do their genome sequence, you know, what is responsible for this. So we don't know. I'm sorry. I'm really sorry. What's your thought on the vaccine working if getting trigger point injection steroids for back problems? So I don't, I'm not a physician, but if it's just locally delivered, and Daniel talked about this today, muscle-delivered steroids that don't go systemic should not have an impact systemically on the immune system, which is in many different locations, right? There are lymphoid organs everywhere. So from a scientific basis, it shouldn't really be an issue. Wide array of opinions. Well, this is not necessarily an opinion. There are websites where you can go and they report the data from the states, right? 59% fully vaxxed. I don't see any reason to think it's it's not an opinion, it's a number. Do you either know offhand or could refer to me to somewhere which describes the changes in locations in spike? You're talking about in the variants? Yeah, go to covariants.org. 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 It's got them all. Should vaccinated people be concerned? No. B1.1.529 has infected 10 people in the world. So for some scientists to say, I'm worried about it, is really disingenuous, in my opinion. And you can find many scientists who are not. So the journalists just found someone who said they were worried about it, and then they ran with it to get attention. Why would we worry? So this British scientist said, yeah, there are like 32 amino acid changes in spike. So what? A lot of the variants have changes. It doesn't matter having a lot. It matters where they are. And we have no indication that this is of any importance. So, I don't know why anyone worries about any of the variants. Just get vaccinated. You're protected. It's SARS-CoV-2. And, these variants were not selected by immune pressure, apparently. They're just arising, and they make more fit viruses, and that most likely has little to do with what happens to us. Why are doctors still recommending ivermectin? I don't know. That happens a lot with a lot of drugs. Some places will still give you hydroxychloroquine, even though azithromycin, an antibiotic. But there is a trial ongoing that Daniel has talked about for ivermectin, and also the other drug whose name is escaping me. But we'll know. This is going to be a good powered, a lot of patient trial, and we'll know in the end what's going on. And we don't have to have. But then, the people who say ivermectin is a miracle drug will not listen to it. They'll say it was bogus. Can you put this on the podcasts? You could just listen at YouTube, right? Why does it need to be in a podcast? You want it to be picked up by your podcatcher? I've only had like 2 people request it. So I don't think it's really worth it for that. But give me a good reason. Because I already have a lot to do, but if people want it, I'll do it. In Gibraltar, they are almost 100% vaccinated. So what are they having a spike in what? Just PCR positives? So that doesn't mean you've transmitted. You may have gotten exposed to virus, and there's still some people who are not vaccinated. Maybe they're in pockets. Lots of reasons for that. You have to be really careful when you see these numbers, because it looks like, oh, wow, vaccine doesn't work at all. But in fact, it sure does, and you have to be careful that you're interpreting the data properly. So I haven't seen the raw data, and I'm very skeptical until I see the numbers. Boosters have the benefit of preventing case in unvaccinated. Yes, it would. But do you want to sell boosters to people by saying you're going to protect the people who don't want to get vaccinated? I don't think that's a good PR story, right? But I do think it would partially protect unvaccinated people, yeah. Thank you, Rob, for your contribution. Really appreciate it. More antibodies, less symptoms, less transmission. I see my spike antibodies go down and get a booster before they called them boosters. A, B in the thousands now. Yeah, Marie, they're in the thousands. They'll go back down. They will contract. That's what they do. Yep. Positive tests matter less the more population immunity you have. I don't know if Amy would agree with that. I understand what you're saying. Once we have very broad immunity, you're not going to be testing because you'll have much less continuous disease. But we didn't do that for other viruses. We didn't do that for any of the previous viruses for which we made vaccines. So not clear to me why you need it. You vaccinate people, you get rid of disease. End of story. What does it matter if it's transmitted, if you get a common cold, if you get it from some unvaccinated person? Well, Amy's gone. No, Amy doesn't have $1,000 shoes, and no, everyone in New York doesn't walk around. Most people wear inexpensive shoes. I see them every day. I commute, and I see lots of people with all kinds of shoes. My shoes are certainly not $1,000 shoes. What type of antibody are made for infections like SARS-CoV-2? Well, first you make IgM, and then you make IgG, and then you make IgA. And the IgA can be in the serum or in the mucosal layers as well. Thank you, Sandy, for your contribution. Really appreciate it. Any thoughts on the hypothesis that NSP14 mutations seen in Japan are causing the virus to mutate to extinction? No, this is ridiculous. If there were a change in NSP14 that ruined the error correction, it would never make it in people. It would never transmit. It is so unfit. It would never make it. So that's a crazy hypothesis. I mean, it sounds good, right? But a less fit virus will not compete. That's why Delta is overcoming everything, has overcome, displaced other variants, because it's fit. It's more fit. And if it became less fit, it would disappear, but there would be other variants that would replace it. So it's a crazy idea that it's going to extinct because of this. It would just be replaced by something else. But you know, before the pandemic, nobody ever talked about this stuff. Nobody cared. Now we have a pandemic. It gets eyeballs in the news, so they talk about stuff like this, which is crazy. Some professor in Japan throws out this crazy theory, which he probably hasn't thought about much, and then the news goes all nuts over it. That's what's happening. So many questions. I don't want to intrude with dumbness. Don't worry about dumbness. Just ask your questions, okay? My philosophy is there are no dumb questions. There are people who are nasty, though, and I don't like them to be here. There's no need for that. We can disagree, but we don't need to be disagreeable. I'm here, so assuming over 1 year protection, will vaccination trigger production of antibodies to both N and S? No. Well, it depends on the vaccine you get, right? That's what I'm assuming. Understanding your question, if you get a spike vaccine, you just make antibodies to spike. If you get infected, you're going to get antibodies to spike and other viral proteins, of course, right? In the future, do you see any potential in mRNA tech to instruct your ribosomes to make more complex things like biologic-type medicines? Have to be a protein, right? So yes, you could design some kind of protein with a therapeutic effect, and maybe it's a novel protein, but then it's going to be recognized as foreign, so it'll get cleared. So you have kind of a race. So I'm not sure about that. Proteins are tough therapeutics. Except for vaccines, they're tough therapeutics. HHS wants to move SARS-2 to select agents. Seriously? Why? SARS-1 is a select agent. Wow. Well, that'll really put a dampener on the good science. This is why, in part, people don't look at infectious virus, because you have to do it in a BSL-3. If you make it a select agent, that means you need FBI clearance to work on it. And that's not impossible, but it's another bar that people might not want to go over. Always a pleasure. Semi-immune compromised 70, wondering if after 2 Pfizers, a J&J boost is good for T-cell durability. Recall Dr. Griffin was going to get J&J boost, but pharmacy said no. Yeah, I don't think that you can do that. But it's an interesting idea because the J&J is an adenovirus vaccine. So if we had a trial to look at that, that would be good. And maybe there is one ongoing. I did a couple of antibody tests and got dimensionless numbers. That's a good way to put it. Yes, it's just a number. 8 and later 20, all the talk of antibodies seems to give numbers with units, helping understand the numbers. So the number you get from the antibody tests, they're in international units, which can be translated to neutralizing potential. But it doesn't really matter because all we know is that 20 is higher than 8, but we don't know if maybe both are protective. Maybe both would protect you against severe disease. Frankly, what clearly protects you against severe disease are the T cells that you have. And you can have, there was a study done a while ago, which we discussed, where they looked for correlates of protection by modeling, and they found that the antibody correlate of protection against any COVID, mild and severe, was, some high level of antibody, which I don't remember, but the correlate of protection against severe COVID and death was very low, which meant to me that it didn't matter. It was the T cells that were doing it. So here's the story. So if you want to be protected against infection, then you need to have high antibody levels. But how high, we don't know. There's actually a paper just come out in Science that looks at different antibody levels in patients and whether they got sick or not. And they say we're defining the correlates of protection. You can take a look at that paper. We'll probably do it Friday on TWIV. But again, it's just a correlate. And I think severe disease has nothing to do with antibodies. I think it's all to do with T cells. So I can't tell you what the numbers mean. 20 is higher than 8. You've got antibodies. You've had 2 vaccines. You have T cells which are not being measured, and they're going to protect you. Should all COVID vaccines be done with aspiration? Daniel Griffin says no. He's the doc here. And I find it hard to believe that you could get all of the vaccine in a blood vessel. A mosquito has a hard time getting its proboscis in a blood vessel. So I just think it's not explaining the myocarditis. What does ACE2 do in adipose cells? I don't know. It certainly doesn't. The adipose cells are not permissive for SARS-CoV-2, if you know what I mean. I don't know what ACE2 does in those cells. Why do anti-core predate anti-surface in a Hep B infection? I think the core is made in a much higher quantity. And that is why you see it. And it may be that it actually comes up earlier in infection. Not quite sure, but that would be my guess. But I do show slides in my lectures where we see that. Yes, that's absolutely right. In controls of cell cultures, are cytopathic effects ever seen in negative controls where everything is the same except no patient sample is put in? Well, you shouldn't, right? That's why you do a negative control, because if you do see CPE, you throw it out and you repeat it because you can't make any conclusions. You did something wrong. You mixed something up. So let me tell you a story. Back in 1981, I had made a full-length DNA copy of poliovirus RNA in a plasmid. And I put it into cells in culture. And after a few days, I took the supernate and I did a plaque assay. And I saw plaques. But I had a control where I didn't put the full-length polio DNA in, and I got plaques on that too. I threw it all out and just repeated it. I must have contaminated something, and the next experiment was fine, and everyone after that, the controls were fine. So if your control isn't what it should be, you throw the whole thing out and repeat it, because that's what controls are for. I very much doubt. So this change, Matt, is A394V. Yeah, this is the kill switch that someone mentioned earlier. If you do that in the lab, the virus can't compete with other viruses. So there's no way that this is propagating in people. Absolutely no way. Oh, look at this. I've been vaccinated for tick-borne encephalitis. That's interesting. The vaccine is used in countries where that disease is an issue. When I find a tick, do I need to deep freeze it to preserve the virus in case I get sick, or is room temperature in a vial enough? Well, tick-borne encephalitis virus is an RNA virus, so it would be good if you could freeze it so that the RNA doesn't break up. Because if they want to do PCR on the tick to see if you've been infected with TBE virus, very interesting. Very interesting. So as far as I understand, in this country, you're still fully vaxxed with 2 mRNA or 1 J&J doses. They still have not said a booster is part of being fully vaxxed. Okay, so it's up to you to decide what you do. I get a text daily from New Jersey saying I can go get a booster. I'm not going. If they make it necessary, I will get it because then I need it to commute and so forth. But that's my understanding. I'm graduating from biotechnology in Costa Rica. I'm in love with virology and molecular biology. I'm considering a master's or PhD in this area. My advice would be to work in a lab for a while and make sure it's what you want to do, because it might not be. So get a job. And if you can't find one, just volunteer and wash dishes in the lab for a while and be enthusiastic and inventive and do things you're not asked to do and show that you love it, and then they'll allow you to do experiments. But definitely work in a lab before you enroll in any graduate program. The data on page 18. I don't know what you're talking about. I haven't seen any data that makes sense for a booster yet, but I'd love to see it. So if you want to send it to me, Doug, send it to me. And people have been sending me stuff that they say makes sense, and it doesn't. It's not good enough for me, but send it to me, Vincent at microbe.tv. You can get memory cells in lymph nodes, sure. They will go there and wait. They circulate also. They go into tissues, but they also go into lymph nodes, and then when you get an infection, they come out after they see antigen in the lymph node. Yeah, for sure. This is a good one. So Truth or Dare loves to make these Saturday Night Live jokes. What's all this fuss I hear about a cardiologist in L.A.? What kind of pervert studies a female rapper? It's great. You had one today or another time in a class. That's great. I love it. We need sense of humor. Things get too intense. What would I recommend against flu? No, having an antiviral is not better than the vaccine. The antivirals don't last 7 years. They last a few days, and that's it. And they're gone. They're metabolized. So if you test positive within 24 hours of symptom onset, you could take Tamiflu or Relenza or Zofluza, different, the two neuraminidase inhibitors and one endonuclease inhibitor. But it won't last forever. The vaccine, it only works 6 months. But I'm telling you, I get a flu vaccine every year. I never get flu. At least not serious flu. And I haven't had a sniffles in a long time. I know when I get a little sore throat and sniffles, but I don't know what it is. But I'm okay with getting flu vaccine every year because the virus is antigenically changing. That makes sense. It's not to boost my antibody levels. It's because the antibodies I have don't match against the prevailing strain. It's a totally different thing than this booster COVID business. Do we know what percent of people have T cells for any common cold corona? So, the number came out last year from Shane Karati's group, right? Something like 17% sticks in my head. How have they fared? So I don't believe they fared any differently, but they do. You can detect these T cells in them in people that have not been infected with SARS-CoV-2. They will cross-react. So as far as making them do better, that's not clear at all. Have you any updates on HIV vaccine? No, not at all. They are putting big money into other approaches like broadly neutralizing monoclonals and approaches to get proviruses out, which are not preventing infection at all. But the vaccines, there have been many, many trials. They've all failed. And names that you have heard of are associated with these trials, like Robert Redfield and the lady Brix, and Tony Fauci, they're all associated with these failed trials. And so we need to try something else. There's no point in doing the same thing over and over. It's just not working. Yes, I should get in trouble, but I don't have a cart. And I don't have a cart to carry it on. And if they want to buy, if EH&S wants to buy me a cart, they can buy me a cart. But I've done this for 35 years. I know how to, I don't really spin them. I twirl them down the hall, but I'm careful. They're not getting out of my control. I'm an old guy. I get to do this stuff, okay? I put caps back on needles. Why? Because I'm really damn good at it. And I'm better than anybody else at putting the cap on the needle. I'm not going to stick myself because I always look at it. I don't look in another direction when I'm putting the cap on the needle. That's what people do when they stick themselves. So now they say, you can't put the cap back on the needle. I'm putting the cap on the needle. Thank you, Mark, for your contribution. Yeah, so I buy my bleach at the supermarket because it's too bloody expensive to buy it from the scientific supply companies. This is $35 a gallon. Out of their frickin' minds. And yes, I get cranky about things. Probably because I've seen too many things and I've seen how things were and how they are now and so forth. In the press, they speculate the third dose might be the last one, but why would that be knowing what we know about antibodies contracting? So, Massimiliano, here's what I think, and Amy's not here because she doesn't agree. There is some thought that, there's this observation that if you are recovered from infection and you get one vaccine dose, then you get a really broad, high-level antibody response that can recognize all the variants, even though you haven't seen them. So your antibody response has the capacity to recognize antigens it's never seen and which don't exist yet in nature, just by the sheer diversity of it. And so they're thinking maybe a booster will approximate what happens when you recover and get a single vaccine dose. Now, Amy would say, and she's absolutely right, that we don't know anything about how that protects you because all they're doing is measuring antibodies. And that's right. We need to have real-world protection studies. But these are getting harder and harder to do now that so many people are getting boosters. And how do you compare it to people without a booster, right? So that's what they're thinking. And they're also trying to make up for the too compressed time in between first and second dose. Shouldn't have been 3 to 4 weeks. It should have been 6 months. But they're thinking they're trying to sell it that way too. So essentially there's no data, but they're worried, so they're preempting it. And I have to say, if there are data that support it, I'll go for it. But right now I don't see it. I feel like I am completely protected against severe disease and death. I'm willing to get a sniffle. There you go. Boy, every week people are into boosters. But I'm glad there's some other questions. Antibodies and the vaccine. Couldn't you argue that things are different because there's a pandemic and boosting antibodies? No, we don't do it for other pandemics. Why are we doing it now? We don't boost in the flu pandemic, which is the only other, HIV, we don't have a vaccine. And so that leaves flu. We don't boost. We just don't do that because we know antibodies contract. So why do we have to do it now? Is there some new vision that we've had? No. If there is, please let me know because I don't see it. Then you guys can get your booster. I'm not telling you not to. In fact, I'm not giving you vaccine advice. My daughter's playing the piano now. Maybe you hear it. I'm just saying why I don't think they're warranted. But what I say is irrelevant because I don't direct any policy. I just am a virologist, who happens to look at it and say, this is what I think. You guys can disagree. It's no problem. But don't say I'm wrong because you don't have the data to say that. Do you find that the push to publish for grant money leads to rushed and sometimes dubious research, even if it's peer-reviewed? Yes, I do. I think the system is a mess at the moment. I think the ones who have a lot of money get more money, and many others struggle, and they shouldn't. The bar is impossibly high. The whole situation with luxury journals being the epitome of publishing, and you should publish in Cell Science and Nature, and then you'll get your grants. This is just ridiculous. This is not how science should be working. Yeah, it's broken, in my opinion. I think you need to change the whole thing, and you should reward creativity, not productivity necessarily. But again, nobody's asking me except you. So there you go. And medical hypotheses will publish just about anything. Isn't it time we started calling natural immunity disease acquired? Yeah, it should have been from the start, but somebody said natural, and a journal picked it up, and boom, it had legs. It's the way this stuff works. What's the data on viral vector vaccines being less effective? So that was a concern at the beginning because we had never used vectored vaccines in this large number, right? So the Ebola vaccine, VSV vector, far fewer people. We were worried that the vector would be an issue, but you know what? It hasn't been. It's really interesting. It's really interesting that it hasn't been. So no impediment. And that's why Russia made, Sputnik with 2 different ad vectors, but it wasn't necessary. Very interesting. What would be a good line of research for long COVID at a mid-sized university with 2 BSL labs? Well, the problem with long COVID is there's no model, right? You'd have to do clinical studies. You'd have to get patient samples and see if you can find biomarkers. And you need temporal samples over time from the beginning of infection throughout for a long time. And it's hard to do in you could do it in a BSL too, but you'd have to have some clinical connections to get those, right? So, since the vector is not replication competent, the vector immunity is short-lived. That seems like a non-answer. I understand what they're saying because the early ad vectors, like ad 5, were replication competent and they did generate more immunity. And so they're not replicating, but viral proteins are being made. So and if you're making an immune response against the transgene, the spike gene, why wouldn't you make it against the vector proteins as well? Unless they're deleted. So let's see. So you're going to give someone a first dose and then you make vector antibodies. You do have the capsid proteins there, but those are not going to be in the vector genome because you don't want to make more capsids. So that could very well be that the vector immunity isn't an issue because of that. They might be right. And I'm deferring because they're the experts in this. I have respect for them and their science. Because you see, Sarah Gilbert said, I think a booster may not be needed. She thought about it and reasoned through the data. Not that she just agreed with me, but I think that's important. Naturally acquired immunity versus vaccine-induced. That's good. Arguably, the COVID pandemic is the world's largest teachable moment this century. And yes, that's why I'm doing this. I'm spending hours every week teaching. I don't mind doing it. I actually love it because I saw it was our opportunity to do this. And I'm growing Microbe TV so we can do it going forward. If development of mRNA vaccine is the greatest lesson learned, what would be numbers 2 and 3? Gosh, that's a tough one. So, yeah, mRNA is the greatest lesson that it works, right? And you can make a vaccine and get it tested and out in people in less than a year. That's really the newest thing. Everything else about COVID we knew. We just didn't know we knew. For example, the contraction of the immune response. We knew that. It's nothing new, but people seem to think it's new. The fact that T cells are important for preventing severe disease, we knew that from other experiences. So what's the science breakthrough? That there's long COVID? No, because we don't understand how that works. And there's certainly sequelae to other viral diseases, so that's nothing new. The loss of smell, no, that happens with other respiratory infections too. So I don't think there's anything else. And people ask me this all the time. What are the great advances of science that we've learned? And really not much. I think there's been a lot of garbage published in all sorts of areas, and because it's shoddy stuff being done too quickly. And frankly, people will tell you they want to save lives, but they also want to get grants. And that's where a lot of the grant money is going. Write a grant to study fiber versus spike. You can't just write a grant. You need to have preliminary data. You're not going to get anywhere just right. You can have the best ideas. You need data. Thank you, Dan, for your contribution. Really appreciate it. New Jersey called you up for a booster? Yeah, I get texts every day. And my wife now is giving me Surus. When are you going to get your booster? So, you guys don't believe me. Neither does my wife. It's okay. I just stick to my science. I got my second Pfizer 18 days after the first. Well, that's pretty close. Sure, why not? It's too close. You boost 6 months out to make up for that too close. Yeah. I think most don't know about memory cells. They think immunity ends once antibodies contract. Yeah, I think that's part of it. And the press doesn't help. They push that forward. But some scientists push it forward also. The cardiologist pushes it forward. Oh, antibodies are contracting. He doesn't use the contracting. He used the W word, but he just pushes it forward without thinking about it. Have you sat on the study section? Yeah, I did for like 12 years. And the last 3 is chair. Yeah. Plenty of memorable reads. No time to remember them now. We only have 15 minutes, but I have to think about it. Yeah, I have so many memorable moments at study section and words that people use over and over again. Study section is such a waste of time. It's such a bloody waste of time. And I know a lot of people are going to tell me you're wrong. Fine, tell me I'm wrong. I was on it for 12 years. You can't tell me that the top 10 grants were really any different and they were any different from the next 10 that didn't get funded. It's just BS distinguishing between them. And I think it's infuriating. Philip is pretty funny. I grew a tail with Moderna, horns with Pfizer, and the 999 birthmark on my head with J&J. Should I be worried? Nah, you're good. You're good. It's funny. You're making fun of some of the questions. I understand. The general public has been made to fear long COVID so much they will never be content with a non-sterilizing vaccine. That's a really interesting idea. Daniel says they have, in his health organization, they've studied now millions of people who have been vaccinated and had infections confirmed by PCR, and he doesn't see any long COVID in them. So it may be that vaccine prevents it for whatever reason. We'll see. And you can get reinfected and you won't get long COVID. So the fears about sterilizing vaccines are possibly unfounded. Yeah. Now, Marie, this is wrong. Antibodies do last 6 to 8 months. And in fact, antibodies can last. You see, this statement, don't count on 6 months. This is offensive. You can find antibodies 40 years after the initial infection. You should take my course because you don't know anything about viruses. Thank you, Thomas, for your contribution. Really appreciate it. Side effects of a vaccine means what? Oh, side effects. So there are a list of side effects that the vaccine manufacturer has going into the clinical trial, like pain at the injection site, muscle soreness, fever, malaise, headache. And then there are things that the people report on top of it. I got run over by a car, stuff like that, right? But there are more serious things. So some of them may have palpitations. Some of them may have a transient episode of not being able to breathe. All of these things, and they all get listed in the trial. And the ones that occur equally frequently in the vaccine versus the placebo group, obviously they have nothing to do with the vaccine. That's what we mean by side effects. Yes, zero cents for lethal mutagenesis. As I've said, people have made that XON change in virus in the lab, and it cannot compete with wild-type virus. So this makes zero sense, and those people never read the literature. So they're just talking out of their you-know-what. Do you know any respiratory or other viruses where we vaccinate after natural infection? Well, let's go through the vaccines for influenza. We vaccinate even if you've had. We tell you to get a vaccine every year, no matter what, if you've been infected or not. We vaccinated everyone against polio, even though some of the kids had been infected, although not paralyzed. We vaccinated everyone against measles. So sure, we do that. Because it's too hard to test people and then decide who to vaccinate and who not to vaccinate, right? If you had to choose to live as a virus or bacteria, which one would you choose and why? Oh, I'd be a virus, of course. They're the most awesome things on the planet. They have minimal genetic material, and look what they can do. They can bring Earth to its knees. Who else can do that? Even the worst dictators lose in the end, hopefully. So virus for sure, because with a small amount of material, they can do so much. Or also nothing. They can be benign as well. And in fact, most of them are benign. For we 80-year-olds, how good is T-cell immunity? The immunity you already have is pretty good, but you can't make new T-cell immunity. That's what you have an issue doing. Wow, these comments are so delayed. Just getting into the Japan business. Thank you, Overjoyed. You too. Everyone have a happy Thanksgiving. Yeah, so ideally 3 to 6 months gives it time for the first one to mature. That's quite clear to me and to Amy. Did cellular organisms evolve from biofilm? I don't think so. I think the biofilm came afterwards, right? Because the biofilm is composed of like bacteria or cells in sheets, right? So I can't imagine that was there first, unless you're thinking of some kind of chemical biofilm, right? Maybe, because that would be protective, yeah? Yeah, that makes sense. I like that. What percent vaccinated worldwide would end the COVID-19 pandemic? I don't know. It depends. So the problem is that vaccination is never homogeneous. There are always pockets of people who are unvaccinated, and that's why you see these big outbreaks. They go to parties in Germany, and you have a big outbreak. So why don't we say combination of vaccinated and infected 80% worldwide, which is not even close yet, right? I think that would do it, in my opinion. That's just me, and you can have your own view. It's okay. The micrograms is what matters, right? The volume doesn't matter as long as you know the concentration. So you should get a certain amount in micrograms with each one. That's what matters. Yeah, that's a good RBG quote, right? That was from Daniel tonight on our update. What if there's common ancestry of the virus precursors within certain populations and therefore innate recognition due to genetic background? I don't quite understand that, but, RNA is sensed, and RNA in viruses is different from in cells in certain characteristics, so they're all pretty much similar. So I don't think there is a common ancestry in terms of the molecular signals that stimulate innate immunity, whether they be RNA or DNA or protein. Yeah, they're all very similar. Yep. Well, so some people want Fauci to go, some people want Walensky to go. I think there have been some problems with messaging, and the CDC has had some data that they prematurely jumped on, like the Provincetown data, which didn't mean what they think it meant. And it's unfortunate. I wish they would just take a little bit longer to think about it. We have 580 people here. If we give us a thumbs up, folks, and get us over 400, let's say, that would be cool, so that more people can find us. Did you both get the second vac 3 weeks? Yeah, 3 to 4 weeks. And are you okay with that? I'm okay because I don't see any reason that it's not okay. I mean, I'm saying that's how they're selling the booster, to make up for the compression, but I'm not sure that that's correct. So I'm out in the world. I'm commuting. I just wear a mask on the train, on the subway, on the street, and otherwise I'm in my office all day, so I'm fine. Last week I spent a lot of time out in New York. I went to a couple of restaurants. I went to a show. I'm protected, folks. But N equals 1 is not science. I understand that. I just feel the science says that I'm good. So all these forums, the problem is it's great if scientists go and comment, but when people and journalists go and then they think this is the final product and this is the message, that's bad. So it's got pluses and minuses having preprints out there. I mean, even published papers have wrong aspects to them, right? And it's hard to dissect those out. So preprints have even more. So this is a problem, and I don't think there is a solution, because I think science needs to be open like that. But I also think more people like me, scientists, should communicate and help people understand what it means, right? Dr. Griffin stated the new Pfizer antiviral is for the unvaccinated only. Correct. Yeah, that's what he said. And I asked him last week, why? Why are they doing that? Do they not have enough? And he said, no, they have enough. I don't remember what but that's correct. It's just for the unvaccinated. But the FDA may have them use it for everyone, right? So I'm getting suggestions for Thanksgiving. I'm just having my family here. We have 3 kids, my wife, 3 kids, and our neighbor down the street. We're all fully vaccinated. My wife and our friend down the street, they both have boosters. My kids don't have boosters. Neither do I. I think we're all fine. And my kids are out and about all the time, of course. It'll be all good. No problem. Mask distancing, ventilation, extra cleanliness boosters all help to reduce transmission. So there's where you're wrong, Tony. How do you know they reduce transmission? How do you know two doses have already reduced transmission? I'm telling you, if you give me a study, it's bound to be wrong, because they're going to look at one time point with PCR, and that's not the way to look at transmission. Sorry, it ain't right. It's not right. So I'm not convinced on the transmission angle either. I support vaccination. I support masking. I support everything. I support everything that works. That's why I'm saying this. How can I explain that vaccines not only reduces likelihood of severe COVID, but reduces the period you're infectious? Well, you say the studies that have been done show reduced PCR loads very shortly after the initial test, which isn't ideal, but it's way below the level you need to be transmitting. That's quite cool. Why didn't the U.S. This is R0, by the way. Hello, R0. Why did the U.S. do attenuated vaccine? Because it takes longer. I was at a meeting of the NIH 2 weeks ago, and they wanted a pandemic plan for different viruses for the future. And they say, no attenuated vaccines, it takes too long. Something like mRNA or something you could make really quickly. Thank you, Copied Often, for your super chat. Really appreciate it. Wow, you're multitasking, huh? How exactly is a virus inactivated? It's treated with chemicals like formalin or beta-propiolactone that maintains the protein in the right structure, so it will make an immune response, but it's no longer infectious. It can't either bind to cells or it can't come apart in the cell. 346 thumbs up. Get us to 400, folks. I'm speeding through some of these questions because it's time, 5 minutes, we need to end this. I don't want to keep you forever. And we'll be back next week. Lateral flow can have different intensity. I don't think that's sufficiently quantitative, Zia. I don't think that would flow. And you couldn't put a number on it anyway, right? And so there's a question about original antigenic sin. I'm going to reserve that because it would take more than a couple of minutes here to talk about. But it's a really good question. If a third dose gave me sterilizing immunity, I would run to get it, but it doesn't, does it? No, it will prevent infection for a couple of months, and then the levels will contract, and you will be infected. How would it impact long COVID? I think 2 doses, as Daniel has said, and you get infected, you're not going to get long COVID. That's his observation. Still early, but he's looked at millions of patients, he said on the last update last week. So I think 2 is fine. And now we're getting just to the part where I said, I complain that people don't listen to TWIV, and it's pretty good. Thank you. I appreciate it. But you guys are all believers. You like it. That's why you're here. You did your marathon training listening to TWIV. That's really cool. Did you do well? I hope so. I love teaching, as you guys know, and I think it's cool stuff we talk about. I'm kind of sad that not more people listen, but that's fine. You guys are here, and I'm good with it. I really am. I do try and figure out how to reach a broader audience, and one of the things I want to try is to make snippets, right, of the TWIVs, break them up. But I got to find some time to do that. I'm packed with what I do now. It's really tough. And so I can't even break out 15 minutes of a TWIV and publish it. I don't have time to do that. 369 likes. Yep. There's so many great questions here. I feel bad every week that I have to skip through them, but I have to. But you guys are having a good chat amongst yourselves, right? Does having recently been boosted increase my chance of a false positive? Well, if you're going to do an antigen test, no. The only thing is, if you're boosted, you're going to be antigen negative, most likely, because you need to get a good infection to have antigen, and I don't think that being vaccinated allows that. Why do I get upset about ivermectin? I don't have a superior. My only superior is my wife, and I don't listen to her narrative. No, I don't. Nobody tells me what to talk about ivermectin. But I have to say, my wife did work on ivermectin at Merck for many years, although she didn't do virus work. I just don't see data that says it works, but I'm waiting for the clinical trial. But nobody tells me what to say. You should know better than that. Come on. That's why I'm arguing with people about boosters, right? Everyone else says you should get a booster, but I'm not. Okay, let's whip through here and find some people to thank. Here we go. Gabriel, thank you. Thank you so much for your contribution. Really appreciate it. By the way, Vanity Nutrition is going to visit the incubator next week and perhaps moderate there from there or check out a pod. So that should be fun. And you guys are in New York, you should let me know. You can come visit, sit in. Thank you, Video 9. Why do some vaccines protect from infection for years like polio, but others contract? Well, they protect against severe disease. So COVID, we don't know if it's a few months, right? Severe disease may be years. But certainly the longest lasting protection against disease are viruses that cause a bloodstream infection, viruses that cause a viremia. And if you look at my lecture, which was about immunity last week, I think, you'll see a table where the longest duration of antibodies, 40 years and more, are in the viruses that cause systemic infection. The mucosal infections are much less, not just months, but they can be years, but not 40 years. Why that is a good question. We actually don't know. No idea. Maybe being viremic, primes the immune response better. Thank you, Martina, for your contribution. Happy Thanksgiving to you as well. RSV vaccine is in the works. There's a mRNA version. The last one was in the 60s, and it made kids have worse disease. So that kind of put a damper on development of an RSV vaccine for many years. Barney Graham was involved with that, figuring out what went wrong with it. He's, of course, very much involved in the COVID mRNA vaccine. And so they figured out what went wrong, and now they're trying the mRNA. But there also is a trial vaccinating mothers, pregnant mothers, and then they will pass antibodies on to their babies because babies are susceptible as soon as they're born, and very hard to vaccinate them at that young age. But getting antibodies passively for 6 months could do the trick before you can vaccinate the baby. So you'll be seeing more about that for sure. Thank you, Ray, for your contribution. Who should get the hepatitis vaccines? Hepatitis B is recommended for everyone in this country. Not everyone gets it. If you work at a medical center like I do, you should get them. If you work with human cells, you should get a hepatitis B vaccine. Infection rates are very high in certain Asian countries, so you should get them there as well. My second Pfizer will be 6 months. I think you're good. As I've said, I think a long interval is great. Will we learn more about the immune system? I do think so. I think we are studying it more intensely than any other time in history with more technology. And even though I poo-poo, us forgetting basic immunology, I do think we're going to learn a lot about antibodies and memory and T-cell and memory and so forth. Yes, I completely agree. But we're not there yet. We are not there yet. And people who say, yeah, we've learned a lot so far, they're just fooling themselves. They're just fooling themselves. Do I believe the main goal for rich people is to centralize all banks into one? I don't think so. I think in some countries, perhaps. Do you think COVID is made up? No, it's not made up at all. I'm a virologist. I know it's real. You have to listen, Sven. You may not want to believe people, but I'm a virologist. This is a real thing, and all the people who say it's fake are just so wrong. I can't emphasize to that extent how wrong they are. You can argue about ivermectin and boosters and all this, but the virus is real. It's killing people, and it's real. No problem. Folks, don't apologize for getting a booster. You absolutely do not, okay? I am just telling you what I think. I've said that many times. What data would you want to see if boosters are needed? I want to see if the rate of hospitalization and severe disease declines, the protection against it declines. Okay, and it has not yet. And I do not see the data that it has in a substantial way. And we'll keep talking about it, okay? Thank you so much, VI, for your contribution. Really appreciate it. I always think of great questions Monday and Wednesday. I can't remember them. Think of them. What were they? Yeah, well, maybe they'll come back. Okay, before I leave you all, let me just see if there's anyone to thank. So the moderators tonight, Vanity Nutrition, Mr. Ozzy, Cam, and Les Fabi, thank you so much for helping out. Really appreciate it. Thank you, Ron, for your contribution. Can it increase PSA? No, I don't think so. I don't see the connection. No. Thank you, Arnaud. I know it's Ro, but I think it's cute that you said Arnaud last time. That's really good. All right, one last thing. So as you know, Microbe TV is now an official 501c3. Hey, 3 more likes over 400. Come on, you can do it. $3.99. So we're a 501c3, which means your contributions to us are federal tax-free in the U.S. So we'd love to have your support, especially here at the end of the year. Next Tuesday is Giving Tuesday, where a lot of people give, right? I'm going to highlight this because I'm going to answer this in a second. So we'd love to get your donations. You can go to microbe.tv/contribute. And as I said, they're tax deductible now. And to make some incentive, here's a big one. If you donate $1,000 or more, you get a free signed copy of Principles of Virology. There's a stack of them behind me next to the guitar. You can't see it. I will sign it and write whatever you'd like me to write. I will not write you need a booster, but I'll write other things. And $1,000 or more, I'll ship it out to you for free. And that's the latest edition of Principles of Virology. And if you can't do that, I understand. But give whatever you can, because we'd love to have help. I'd like to hire people next year to help out and make more great science content. Can you please recommend a good immunology text? So if you want a basic text, How the Immune System Works by Lauren Sompyrak, right? If you really want really, really basic, I use Janeway's Immunology, which is really, really, really advanced, but it's got everything in it. So there are two extremes there. Okie dokie. We did 414. Thanks, folks. Really appreciate it. Everyone, have a great Thanksgiving if you celebrate it. If you don't, have a great weekend. Be safe and store up your questions for next week, because Amy and I will be back next Wednesday. And, of course, Monday we will resume Virology Live. I so appreciate having all of you here, folks. Thanks for coming. We'll see you next time. Good night.